Well, this video is about clinical science from Mrcp Part 1. It's a very important side, one of the most important ones, because a lot of questions are coming from here, 25 questions at least. Will be coming from here. It's a vast subject as well, so we need to cover up all these things. Cell organelles, endoplasmic reticulum. What's the function? It's translation and folding of new proteins. Rough endoplasmic reticulum. That's the function of rough endoplasmic reticulum, and expression of lipids would be function of smooth endoplasmic reticulum. So protein rough and lipid smooth. Mitochondria is it's the powerhouse of the cell. Energy production contains mitochondrial genome as circular double-stranded DNA. Not sure. And then Golgi apparatus, sorting and modification of proteins, very important as well. DNA maintenance and RNA transcription happens in the nucleus. Lysosome breakdown of large molecules such as protein and polysaccharide, and nucleolus ribosomal ribosome production. Ribosome translate of RNA into proteins, also very important. Peroxisome breakdown of metabolic hydrogen peroxide. Proteasome, along with lysosome pathway, degradation of large protein molecules, breaks down protein molecules. The cell cycle, also very important topic. What happens is M, that is mitosis, cell division, M phase. So mitosis or cell division, this is the M phase. G1 is gap phase 1, determines length of the cell cycle under the influence of p53 enzyme. S is for DNA synthesis, and G2 is gap phase 2. So duplication happens here, separation happens here, and this gap. This book is actually giving a brief, not brief actually, to the point stuff, like things that you really need to know. These are not in detail, so for details you need to go through some other big notes maybe. Membrane receptor, there are four main types: ligand gated ion channel, tyrosine kinase receptor, guanylate cyclase receptor, gibberellin coupled receptor. So each has its own thing. Ligand gated ion channel, basically example is nicotinic acetylcholine, GABA A and C glutamate receptor. All these are ligand gated, generally mediate fast responses. Okay, tyrosine kinase receptor, like insulin, growth factor, interferon, contain intrinsic enzyme activity. And then guanylate cyclase receptor contain intrinsic, is similar to this one, contain intrinsic enzyme activity. ANP nitric oxide receptors. And then the most important one, as in the most common one, gibberellin coupled receptor, example muscarinic acetylcholine, adrenergic receptor GABA B. So generally mediate slow transmission and affect metabolic processes, very important. Activated by a wide variety of extracellular signals like peptide hormones, biogenic amines, lipophilic hormones, and light. Consists of three main subunits: alpha, beta, and gamma. So also very important. Ligand binding, conformational changes to receptor. This induces exchange of GTP for GTP. Okay, now going to the adrenoreceptors, that is basically the ones that we're talking about: alpha, beta, and gamma. So alpha 1, there's agonist and there's antagonist. So alpha 1 agonist would be phenylephrine, an example, causes vasoconstriction. So you have to remember that alpha 1 causes vasoconstriction. Phenylephrine is an example. Relaxation of GI smooth muscle also happens. Salivary secretion also happens. Hepatic glycogenolysis, so glycogen breaks down to glucose. Have to remember this. And then alpha 2 agonist is clonidine, mainly presynaptic inhibition of transmitter release. Noradrenaline or acetylcholine from autonomic nerves inhibits insulin platelet aggression. Okay, then beta 1 agonist. So you also have to remember one thing, that alpha 1 agonist has in vasoconstriction, so it will increase blood pressure. So alpha 1 antagonist is the one which will use for decreasing blood pressure. Beta 1 agonist, and this selectively for the heart. Beta 1 agonist, like dopamine, mainly located in the heart. Beta 1 receptors are only in the heart, so it will increase heart. An agonist will increase the heart rate and heart contraction force. So beta 2 agonist is like generalized, not only in the heart, generalized. Agonist is salbutamol, which we use for vasodilatation. The inhalers that we use, salbutamol, bronchodilation, relaxation of GI smooth muscle. Okay, beta 3 agonists being developed may have a role in preventing obesity. Lipolysis can happen. So these are all very basic stuffs which we all need to know, and very important stuff as well. So pathways will be all RG protein. Okay, for pathways, all RG protein coupled, that is alpha 1 to 3. Alpha 1 activate phospholipase IP3DAG. Alpha 2 inhibit adenylate cyclase. B1 stimulate adenylate cyclase. B2 same. B3 same. Secondary messengers allow amplification of external stimulus in cells. So cyclic AMP, protein kinase activity, cyclic GMP, calcium, all these things, adrenaline, all these things that they, if you remember this, protein kinase would be insulin growth hormone. Yeah. Molecular biology techniques. This table shows a very summary of techniques. Southern blotting, these techniques detects DNA. Northern blotting detects RNA. Western blotting detects and quantifies protein. So what you have to remember is this small box, which will help you to remember all of them. Snowdrop, south for DNA, north for RNA, W for protein. So all these. Now comes polymerase chain reaction. It's a molecular genetic investigation technique. The main advantage of PCR is its sensitivity. Only one strand of sample DNA is needed to detect a particular DNA sequence. It now has many uses, including prenatal diagnosis, detection of mutated oncogenes, and diagnosis of infections. PCR is also extensively used in forensics. Prior to the procedure, it's necessary to have two DNA oligonucleotide primers. These are complementary to specific DNA sequence at either end of the target DNA. So, they've also mentioned preparation for PCR. There is initial prep. Sample of DNA is added to the test tube along with the two DNA primers. A thermostable DNA polymerase TAQ is added. The following cycle is then takes place. The mixture is heated to almost boiling point, causing denaturing of the DNA. Okay. Then mixture is then allowed to cool. The complementary DNA strands pair up. As there is an excess of primer sequences, they pair up with the DNA preferentially. The above cycle is then repeated with the amount of DNA doubling each time. So that is PCR. And then there's reverse transcriptase PCR, is to amplify RNA, like in COVID-19. RNA is, we do RT-PCR for COVID. Reverse transcriptase PCR. RNA is converted to DNA by reverse transcriptase. Gene expression in the form of mRNA rather than the actual DNA can therefore be analyzed. This is for amplifying RNA. Okay, the next thing would be anatomy. So this is not really like very much in details or anything. This is just the important things that basically you need to know for Mrcp part 1. Simple details, not much, and helpful as well. So foramen of the skull. So when we talk about the skull, we have to see pictures, and it's very important as well. So this is like the base of the skull. Some things which you need to know is A, that is optic canal. What are the things associated with these structures? That's all you need to know. And so A would be this one. This is from the base of the skull, and this is like the eye socket. A is the optic canal. The bone which is involved is sphenoid. Most of it is sphenoid here, nearby the optic canal. Vessels that go through it is ophthalmic artery and optic nerves for that. Superior orbital fissure, that is B. This one, this B is here. The bone involved is sphenoid again. Superior ophthalmic vein and inferior ophthalmic vein, not artery. The nerves that go through these are oculomotor nerve, trochlear nerve, lacrimal, frontal, and elastociliary branches of ophthalmic nerve, abducens nerve, many nerves. And then comes the inferior orbital fissure, that is sphenoid and maxilla are like involved in it, and vessels going through it, inferior ophthalmic vein, infraorbital artery, and infraorbital vein. Okay, the nerves, zygomatic nerve and infraorbital nerve of the maxillary nerve, orbital branch of pterygopalatine ganglion. Then there is foramen rotundum. Bone involved is sphenoid, maxillary nerve P2. Foramen oval, that is E, which one here? Sphenoid, accessory meningeal artery, mandibular nerve. Okay, jugular foramen, that is the big one. This one, this foramen magnum. So jugular foramen is this, the next to it, F. Also occipital and temporal, not sphenoid. Posterior meningeal artery, ascending pharyngeal artery, inferior petrosal sinus, sigmoid sinus, and jugular vein. And there are very important nerves going through it: glossopharyngeal, nerve, vagus nerve, external nerve. So these things we need to have like an average idea of what goes through what. Like superior orbital fissure 3, 4, 16 important nerves, 9, 10, 11 for jugular foramen, and 2 for optic canal. Okay, this is something which keeps on coming, and if you are not really paying attention, you make mistakes. Renal anatomy. The table shows the anatomical relation of the kidneys. So for the right kidney, as you know, this is the right kidney, and then this is the left kidney. Right kidney, right suprarenal gland, that is adrenal gland. This is duodenum, then there is this colon, and this is direct contact, and layers of peritoneum in between liver and distal part of small intestine. And the direct contact with left kidney would be suprarenal gland again, pancreas and colon. So this is very important. Layers of peritoneum in between stomach, spleen, and distal part of small intestine. Okay. Next is skin, layers of the skin. The layers of the skin. Epidermis is the outermost layer of the skin and is composed of stratified squamous epithelium with an underlying basal lamina. It may be divided into 5 layers. So the 5 layers, if you remember, is like C, L, G, S, G. Okay, from the outside to the inside. C, L, G, S, G. You can write it down to remember. So stratum corneum, flat dead-like scale-like cells filled with keratin, continually shedding. So of course, it's the outside layer; it keeps on shedding. Stratum lucidum, clear layer, present in thick skin only. Stratum granulosum, cells form linked with neighbors. Stratum spinosum, squamous cell begin keratin synthesis. So here's where keratin starts to grow. Thickest layer of epidermis. Stratum germinatum, the basal membrane, single layer of columnar epithelial cell, give rise to keratinocytes and contain melanocytes. So spinosum, keratin, germinativum, melanocytes. Need to remember that. Okay, now hypersensitivity, also a very important topic. This falls under immunology, and some important topics are there for immunology. Okay, for immunology, hypersensitivity, what are the types?